MS Ramaiah University of Applied Sciences.
Determination of differential leukocyte count. Aim of the experiment: to count and calculate the different types of leukocytes present in blood sample and report, and to study the significance of differential leukocyte count in diagnosing the diseases. Introduction. Many hematological and other disorders can be diagnosed by a careful examination of a stained blood film. Differential leukocyte count along with total leukocyte count helps to detect infection or inflammation, determine the effects of possible poisoning by chemicals, drugs, chemotherapy, radiation, etc. Differential leukocyte count is also done to monitor blood diseases like leukemia or to detect allergic or parasitic infections. The purpose of this experiment is to assist the diagnosis of diseases in relevance to an increase or decrease of a particular type of leukocyte. Experimental setup: compound microscope, 5 to 6 grease-free clean glass slides, Leishman stain, pasture pipette or glass dropper, distilled water, blood lancet, cotton swabs and 70% alcohol. Experimental procedure: preparing the blood films. Get a sterile finger prick, discard the first drop and allow a medium sized drop of blood to form. Place a small drop of blood about 2 cm from the end of the slide. Without delay, place the spreader at an angle of 45 degrees to the slide. Move the spreader slide to touch the drop of the blood. Drag the spreader slide to the other end of the slide by a single smooth, fairly fast gliding motion with uniform pressure. Air dry the smear formed. Fixing and staining the blood film. Place the slide on a staining rack assembled over a sink. Pour 8 to 10 drops of Leishman stain on each unfixed slide using a dropper. Allow the stain to remain undisturbed for 1 to 2 minutes. Later, add equal volume of distilled water to the stain carefully without spilling over. Mix the stain and water by gently blowing at different places on the slide with a dropper without scratching the smear. Allow the diluted stain to remain on the slide for 6 to 8 minutes. Flush off the diluted stain in gentle stream of distilled water for about 30 seconds. Drain the slides facing downwards. Blot the undersides of the slides with filter paper and air dry. Identification of leukocytes under oil immersion lens. Place a drop of cedar wood oil in the center of the slide and focus under oil immersion lens. Identify the different types of leukocytes from its size, nucleus color, cytoplasm, nucleus ratio and staining properties. Precautions: absolutely clean, grease and dust free slides must be used. Do not allow the Leishman stain to become syrupy or dry up, as it is likely to cause precipitation of the stain. Avoid using tap water. Keep Leishman stain in well stoppered bottle after use to avoid evaporation. Avoid repetition in counting. Avoid counting the cells in extreme ends and edges of the blood film in the slide. Hello students, today we are going to study the determination of WBC count. The aim of this experiment is to count and calculate the number of WBCs present per mm cube of given blood sample, and also to study the significance of WBC count in the presence of various diseases or disorders. WBC or white blood cells act as major defense system or first line defense system in case of invaders like bacteria, pathogens, viruses. WBC count is kept remarkably constant in healthy individuals maintaining their immune system. But the WBC count increases or decreases in many disease conditions such as leukemia, allergies, infections and other disorders. Now let us start the experiment by studying different components which are utilized in this experiment. The different components used in this experiment are blood lancet, which consists of a sharp needle to prick the finger bulb. Lancet device which is used to place the lancet cotton swab with 70% alcohol is utilized to sterilize the area. Dry cotton swab to remove the excess of alcohol. WBC pipette which contains stem, rubber tube and mouthpiece. The stem has a graduation of up to 20 microliter. Neubauer's chamber with cover slip. The Neubauer's chamber is a single solid heavy glass slide. The floor piece is divided into two equal parts by a short H-shaped trench in its middle. Identically ruled areas called counting grids containing of squares of different sizes are etched on each floor pieces. The counting grid has following dimensions. WBC diluting fluid, or also called as Turk's fluid. It consists of glacial acetic acid which hemolyzes the RBCs without affecting the WBCs. It contains gentian violet which stains the nuclei of WBCs, and it also contains distilled water. For experimental convenience, we have transferred Turk's fluid to a smaller container and compound microscope. Now let us start the experiment. First, focus the Neubauer's chamber under low magnification, that is 10x, and identify WBC counting areas. Neubauer's chamber is collected and focused under low magnification, that is 10x, and illumination is adjusted such that the WBC counting areas are focused and seen. Now you can see the WBC counting area without changing the focus or field of magnification. Remove the Neubauer's chamber and transfer it to the working area. After transferring the Neubauer's chamber to the work table, cover the counting grids or counting area with a clean cover slip in such a way that it covers the counting areas on both the sides of the smooth surface, and the Neubauer's chamber is kept ready for charging. Then sterilize the finger bulb with cotton swab containing 70% alcohol. Allow the alcohol to evaporate. Then, using the lancet device, take a bold prick. Using the dry cotton swab, wipe the first drop of blood as it may contain debris. Gently squeeze the finger bulb and allow a clear drop of blood to form, and then using the WBC diluting pipette, suck the blood up to the mark of 0.5. Care should be taken, there should not be any air bubble entrapment, and if in case excess blood is drawn, then it is removed by touching the tip of the pipette to the cotton. Now as you can see, the blood is drawn up to the level of 0.5. Now dilute it with diluting fluid, that is Turk's diluting fluid, up to the mark 11. Now as you can see, the diluting fluid is sucked up to the mark of 11. Then gently put a knot to the rubber tube to avoid any leakage of fluids while mixing the contents. Place the tube horizontally between both the palms and with a gentle horizontal motion, mix the contents thoroughly for 3 to 4 minutes. After mixing the contents for 3 to 4 minutes, open the knot and discard the first 2 to 3 drops of fluid, as it contains only the Turk's fluid but no cells. Then allow a small uniform droplet to be formed at the tip of the pipette, and this droplet is charged onto the Neuber's chamber. Avoid overcharging or undercharging of Neuber's chamber, and care should be taken. There should not be any excess fluid in the H canal of Neuber's chamber. After charging, the Neuber's chamber is allowed for settling down of cells for 3 to 4 minutes. After 3 to 4 minutes, transfer the Neuber's chamber to the stage of compound microscope, and it is observed under low magnification, that is 10x. Now after 3 to 4 minutes, we are transferring the Neuber's chamber to the microscope. Now focus the counting area, that is WBC counting area, and use the fine adjustment to identify the WBCs under low magnification. The WBCs appear as round, shiny and darkish dots with a halo round around them. These dots represent the nuclei stained by gentian violet, and the cytoplasm is not stained. Now as you can observe, the WBC cells can be seen. Now count the cells in 4 corners, or the 4 corner squares, employing the counting rule. Switch over to higher magnification if required to observe the cells. This completes the determination of WBC count.
At MSR UAS, we spark your imagination unlike any other.